Hi everyone, my name is Nur Amalina binti Ali Hashmi with metric number 195836. So today, I would like to do a tutorial on the pairwise sequence alignment, which focusing on practical 2.1 and sequencing results into full-length UPM1 lipase gene. So basically, we want to obtain a full-length UPM1 lipase from a pair of primers. Earlier, these two primers were designed based on the UPM1 lipase gene sequence, which have been cut by restriction endonuclease, MS1, and ENCO1. So first of all, we have to open BLAST using this link. Then it will appear like this. We click on the nucleotide BLAST. Okay. Then it will appear like this. So first, we will paste our forward sequence in our practical at this query sequence box. Okay, we want to copy this one, but we have to remove the end at the beginning and also end of this sequence. We don't want the end. Why we have to trim the end? Because the end is scored as misalignment, which cause the sequence to appear to be less related to the reference sequence later in the BLAST. So we click this one and copy. We open the BLAST and we paste here. Okay. Then we have to click this box, align two or more sequences box. Now we have to open our practical and copy the reverse sequence. We will do the same, just like the previous one. We have to trim the end at the beginning and also end of our sequence. So now we copy and we paste here. Then we click BLAST. Now it will appear like this. This means that our sequence has a match in the BLAST, which is we click alignment tab, which is the sequence ID Curie 50875. So it matched with our sequence. So we want to look at the identities here. Now we will use the bioedit program to combine both of the forward and reverse primer to obtain a full-length UPM1 lipase. So after we open the bioedit program, we click File, New Alignment, and it will appear like this. We click Sequence, New Sequence. For the name, we put the name of our first primer, which is the forward primer, and we select DNA at the sequence type. At the box here, we have to paste the sequence of our forward primer. But remember to trim the end at the beginning and also at the end of the sequence. So we paste here and we click apply and close. And we will see a sequence like this. Then we will do the same thing for the reverse. Sequence, new sequence. The name is reverse. Sequence type, we select DNA. The same thing, we copy the reverse sequence without the end. Then we click apply and close. Then we will forward and reverse sequence here. But now, before we can combine both of them, we have to reverse the sequence of this reverse primer. Why? Because we want to invert reverse sequence so that they will align with the forward sequence in order to combine both of them. So we click the reverse. We click sequence. And then we click nucleic acid. Then we click reverse complement. Okay, now after we have reversed the sequence of the reverse primer, we have to choose both of the forward and reverse to combine them into a full length. So now we click accessory application, we select cap contig assembly program, and we click run application. So for this dialog box, we exit the dialog box and it will appear like this. So at the end, you should only get one sequence of this contig sequence. So that's all for practical 2.1. Thank you.